This is the super fast run through of the B3 unit. So let's begin with gas exchange. We're going to jump right in. Okay, all organisms do gas exchange. When you are a unicellular organism, you can do it directly right through your membrane. But if you are multicellular, you're going to have a larger amount of body that a volume of their body that needs to be provided with those gases and those waste gases removed, and you're not going to have enough surface area to do so. And so we're going to see that we have adaptations of multicellular organisms to be able to do gas exchange more efficiently. Now, all gas exchange surfaces are permeable. They have a large surface area, they're moist, and they are very thin for a short path of diffusion. And so we're going to see that there's a higher concentration of oxygen in the atmosphere than in deoxygenated blood that's passing by through the capillary, passing by this alveoli. And so the oxygen is going to diffuse down the gradient naturally. No ATP needed. It's passive transport through this very, very short path of diffusion. Okay, now the lining of the alveoli must be moist so that that gas, the oxygen gas, can become dissolved oxygen as it meets with the moist surface of the alveoli. Okay, similarly, carbon dioxide is just diffusing down the concentration gradient because there's more concentration of CO2 in this blood that's arriving at the alveoli than there is in the atmosphere. And so it just diffuses out, and then this can be inhaled, exhaled. Same thing goes for a plant cell. We have this spongy mesophyll layer, which is adapted for gas exchange. It's moist, thin tissue layer, large surface area because of all of these gaps, and permeable. All right, so again, a small organism can respire directly across. Large organisms are limited because they don't have that same large surface area compared to volume, and so we have special organs that are adapted for gas exchange, in our case, lungs. Okay, fish, on the other hand, have gills. They are constantly sucking in water through their mouth, and that water runs across their gills, where again, we have passive diffusion of oxygen into the fish directly through the gills and the capillaries that line them. Okay, here we have the trachea, sometimes known as the windpipe. It then branches into two bronchi. Those bronchi further branch into bronchioles, which then end in alveoli, which look like these little clusters of grapes that have a large surface area. Think about how much more surface area there is on each inside of each of these grapes compared to, say, if this was like one solid mango-shaped fruit, for example. So that is an important adaptation. Okay, altogether, this is the thorax or the thoracic cavity. Okay, adaptations of mammalian lungs for gas exchange. So we have that branched network of bronchioles that's going to lead to alveoli. Again, they have that high surface area. If we were to just have our skin and do gas exchange directly across our skin, like a unicellular organism would do it directly around its outer membrane, we would need to have 40 times the surface area to support the amount of volume in our bodies. And so it's really important we have those lungs with alveoli providing that 40 times surface area. Okay, we have extensive capillary beds surrounding those alveoli. That allows for that easy exchange from the air in your lungs into the bloodstream. Okay, and we have surfactant that lines the alveoli. Remember, that's made by those type 2 pneumocytes. That reduces the surface tension and prevents the alveoli from collapsing when you exhale. Okay, speaking of exhale, in science we're going to use the word expiration, breathing in as inspiration, and together we call it ventilation when you breathe in and out. Note that the trachea and bronchi, okay, have cartilage in their walls to ensure they remain open. Cartilage is like what's in your nose and your upper ear. Okay, the bronchioles have smooth muscle fibers, which allows the width of those airways to vary instead. All right, now gas is just always going to move from high pressure to low pressure. And so if you picture your lungs, when you inhale, the volume of your lungs increases. That lowers the pressure of the air inside the lungs. Therefore, the air outside in the atmosphere is a higher pressure, and it's going to move from high to low into your lungs. Okay, vice versa, when your lungs contract, that is going to put that air in your lungs at higher pressure, and air moves from high to low, so now air is going to move out. Okay, now how does that change in volume occur? Okay, it occurs through two sets of antagonistic muscles. First up, lining the outside of your rib cage are these external intercostal muscles. And when they contract, they pull your rib cage up and out, increasing the volume of your lungs. Just picture that when they contract, they're pulling in that direction. So externals contracting, pull it outwards, larger volume. And it's antagonistic with the internal intercostals on the inside of the rib cage. And so when the externals contract, the internals are going to relax, and vice versa. Okay, when the internals contract to lower the volume of the lungs, the externals are going to relax. Okay, you have another set of antagonistic muscles, which is the diaphragm and the abdomen wall muscles. Now, the diaphragm is relaxed in a domed state, as you can see here. And so when it contracts, it's going to contract down, which then increases the volume. At the same time, when it contracts, the abdomen wall relaxes. Vice versa, when you breathe out, the diaphragm is going to relax back into that dome position, decreasing the volume. Okay, and that happens when the abdomen wall muscles contract. Okay, so it's important to be able to discuss inspiration and expiration, what's happening with each of these sets of antagonistic muscles, and how that then affects the volume, and therefore how the pressure of the air going from high to low is going to cause air to move in or out of the lungs. Now we can measure this using a spirometer. Okay, when you are just breathing in and out, if we look at the volume of air that comes in and out for each breath, we call that the tidal volume. If, though, you take the biggest breath you can beyond just at rest, we call that amount that you can take in the inspiratory reserve. Okay, similarly, outside of normal at-rest breathing, we have an expiratory reserve if you blow out as hard as you can. And together, if you add up the inspiratory reserve, the tidal volume, and the expiratory reserve, that would be what we define as your vital capacity. But even if you try to breathe out all the air you can, you're still going to have air in your lungs that you cannot fully get out. And we call that the residual volume that stays in your lungs. All right, let's switch gears now to talk about plants. So leaves have this spongy mesophyll layer, which is adapted for gas exchange. Okay, the lower epidermis is going to contain stomata that are surrounded by guard cells that can open and close this space. Okay, when it's open, that allows for oxygen and CO2 to diffuse in and out, as well as water to transpire out. Now remember, the top layer of palisade mesophyll is adapted for photosynthesis. Okay, it's got this column-like shape like a palisade, which allows for the maximum number of cells to receive light energy. Okay, but that spongy mesophyll lower layer is going to have that large surface area. Okay, it's going to be moist. It's going to be available for gas exchange. Now, also up on the surface of the leaf is going to be a waxy cuticle. Okay, that's going to help prevent water loss. The epidermis are these outer layers of the leaf. There's air spaces within the spongy mesophyll that has a large surface area for gas exchange. Our stomatal guard cells, okay, those are going to close at night when photosynthesis isn't occurring, so the plants don't need a CO2 source. Okay, or when there is water stress, and so we want to avoid having water transpire out. Okay, there's also going to be veins that include xylem and phloem vascular tissue for movement of water and sugars. Okay, this is a drawing skill. It's a plan diagram, so instead of drawing individual cells, your job is to just draw the tissue boundaries. And so you would draw your upper epidermis, your lower as fairly thin regions, and then both of your mesophyll layers as thicker regions, and then your xylem and phloem, xylem up here in the palisade mesophyll, phloem down below. All right, let's talk about transpiration. Now, there are two major factors that affect transpiration rates, and transpiration is just a fancy way of saying the evaporation of water specifically from leaves. Now, there's a positive correlation between temperature and transpiration, which means that as temperature increases, transpiration also increases. At higher temperatures, there's just more energy available for evaporation, and warmer air can hold more water vapor before becoming saturated. On the other hand, there's a negative correlation between humidity and transpiration. As humidity increases, transpiration decreases. And this makes sense. The more water vapor in the air, the less of a concentration gradient there is between the leaf and the air, and so water is not going to transpire out of the plant as quickly. Okay, again, that transpiration, okay, is going to be controlled using guard cells in the stomata. Okay, now if we wanted to count the number of stomata, okay, some leaves, you can actually peel that lower epidermis right off. Okay, you could put it on a microscope slide with a drop of water and take a look and count how many stomata there are. Okay, but not a lot of plants can peel that lower epidermis that easily. Another technique that works for some plants is painting a colorless nail polish on the bottom of a plant leaf, letting it dry, and then peeling off that layer of nail polish, putting it on some clear, transparent tape, and then placing that under a microscope, and then being able to count the stomata. Now, our stomatal density is just the number of stomata divided by the area of the field of view. All right, at this time, that's the end of SL content for B3.1. Okay, we're going to move into HL now, but we'll come back to SL with B3.2 in a bit. All right, hemoglobin is the oxygen transport protein carried by red blood cells. Remember, it has this quaternary structure because it's made up of 4 polypeptides. Okay, and it's conjugated because it has 4 heme groups in addition to the polypeptides. Now, each of these heme groups can bind to 1 oxygen. So a hemoglobin can bind to 4 total oxygens. Now, we're going to see, okay, that cooperative binding occurs because when 1 oxygen binds to a hemoglobin, that is going to increase the affinity for oxygen binding of the other 3 heme groups. And so 1 is going to bind and the other 3 are going to follow soon after. The reverse is true as well. When 1 oxygen is unbound or is released, okay, the other 3 are also going to go very quickly. Now, why is this helpful? Well, because that hemoglobin is traveling past the lungs very quickly. If 1 oxygen binds, okay, and if we were to go at just this proportional pattern, right, the red blood cell might already be past the alveoli by the time another oxygen decided to bind. And so by having 1 oxygen bind and then the rest cooperatively bind more quickly, that allows for more efficient pickup of the oxygen as well as distribution of the oxygen when we get to a cell that needs it. Okay, the next thing you should be familiar with is maternal blood does not directly flow through a baby. That's kind of a misconception that a lot of people have. Those blood systems are completely separate. They might be different blood types. If the mother's blood was flowing through the baby would start to attack it as non-self and vice versa. Okay, so those blood systems are completely separate. And instead, what we have is a placenta that separates those blood systems and just allows for transport of the important materials. So it allows for oxygen to transport from the mother's blood to the baby's, CO2 to transport back, right? Glucose and amino acids to baby, urea and other waste products back, etc. So it's sort of that in between that allows for transport of materials without the blood directly mixing. So the question is, how does oxygen move from the maternal hemoglobin to the fetal hemoglobin? And it turns out humans produce different types of hemoglobin before and after birth. And that fetal hemoglobin that's produced before birth has a stronger affinity for oxygen than adult hemoglobin. Therefore, oxygen will dissociate from the hemoglobin in maternal blood and bind to the hemoglobin in the fetal blood. Okay, and then lastly, we have the Bohr effect or the Bohr shift. How this works. When there is increased CO2, for example, during exercise, or if a cell just did respiration, increase of CO2 lowers the pH. Just think of ocean acidification. You know that increasing levels of CO2 in the atmosphere are making the oceans more acidic, having a lower pH. So we're going to see that whenever CO2 interacts with water, that's going to increase the acidity. But the effect of reducing the pH where there's high levels of CO2, okay, is that hemoglobin then has a reduced affinity for oxygen, which is great because then wherever CO2 is being produced, more oxygen is going to get dropped off. Okay, for example, in the lungs, the pH is 7.4, while in active muscle, the pH is lower at 7.2. So even that small difference, that slightly more acidic pH, an active muscle is going to cause oxygen to be dropped off there, okay, when it gets picked up in the lungs. All right, this is just another summary then of those three important things. You should be able to describe that S curve that's due to cooperative binding. Okay, you should be able to talk about the S curve for fetal hemoglobin being further to the left, indicating an increased affinity for oxygen so that oxygen travels from mother to fetus. And lastly, the Bohr shift. Okay, when you have two different concentrations of CO2, okay, the curve for higher CO2 is displaced to the right, showing a decreased affinity. So remember, the one closer to the left is going to show increased affinity. Okay, and so here we're going to show greater release of oxygen from hemoglobin at the same partial pressure of oxygen. Moving on now, we are going to head over to B3.2 and talk about transport, both in animals and plants. First up, we are going to see that our blood vessels moving away from the heart are called arteries. A for artery, A for away. So arteries move away from the heart. Veins move into the heart. There's an IN hidden in vein. Veins go into the heart. Now, a lot of misconceptions are around whether it's oxygenated or deoxygenated, and that does not have anything to do with whether we call it an artery or a vein. It just has to do whether it's away or into the heart. Okay, they can be both oxygenated and deoxygenated, as we will see. Now, arteries and veins are connected by a network of capillaries. Okay, these are extremely thin, and we're going to talk about some adaptations because capillaries are where gas exchange and material exchanges occur. Okay, so what are those adaptations for capillaries? They have a large surface area. There's a lot of branching. They're very narrow. Okay, capillaries can be as thin as just allowing for a single red blood cell to be able to pass through it. Okay, but those capillaries are going to be near every single cell in the body in order to be able to provide them with oxygen and nutrients and pick up CO2 and waste. Capillary walls are extremely thin, okay, allowing for easy diffusion because of the short path of diffusion. Okay, and then some capillaries have fenestrations. This is like, if you take French, the word for window is fenêtre. It's the same type of root there. Those windows in the capillary that allow for fast exchange of materials under certain conditions. Now, you may be asked to identify an artery and a vein in a micrograph, and there's some things that you should look for. First of all, arteries tend to have a thicker wall. Remember, arteries are moving blood away from the heart, and so when the heart contracts, that blood is moving out at high pressure. That's a glob of high pressure blood, and that artery is going to need to be elastic and be able to stretch as that big glob of blood moves through it. Okay, veins are where the blood is returned to the heart. And so the blood is no longer under that high pressure. It's draining out of the capillaries and into the veins. And so we're going to just have a thinner wall. Okay, arteries tend to be circular in section, meaning if you took a cross section. Veins can be circular or they could be flattened or other shapes. Now again, the artery is going to need to stretch. That's what your pulse is. Your pulse is globs of blood that are passing through arteries as they move away from your heart. And so having this inner surface corrugation, this back and forth, that allows for it to stretch. Veins aren't going to need that because veins don't have a pulse. The blood is just draining out of the capillaries all at once, and so they're not having pulses of blood move through, and so we don't need to be able to stretch. Okay, similarly, then reasons, we have very visible elastic fibers. In veins, fewer or none. So if you're provided a micrograph, you would need to be able to identify this as an artery, this one as a vein. Okay, and sometimes be careful because arteries may not be exactly circular. This one is an artery. You can see the thicker wall and the corrugations, but it has been quite a bit flattened, whereas this one is the vein. So just watch for all the clues. Okay, arteries are going to have a relatively narrow lumen. This helps it to maintain high blood pressure and high velocities or speeds of blood flow, because that's where the heart is directly pumping blood into. Okay, now in the textbook, we have these 3 layers of artery walls. I don't think you would ever be asked about them. I've never seen a question about them, but here they are if you want to have something else to memorize. Okay, you should be able to find the pulse, either right here, if you put 2 fingers right under your ear and slide them down your jawline, you'll feel a pulse there. Use your 2 fingers, okay, or you can find one on your wrist in this location. Okay, don't use your thumb because the thumb actually has a pulse, and that can cause confusion when you're trying to count the heartbeats. Okay, let's talk about adaptations of veins. Remember, veins go back into the heart, and so they're going to have valves to prevent backflow because they're moving a lot lower pressure. And so when the valves are open, that allows for unidirectional blood flow. If it tries to flow back, the valves will close and it will prevent backflow. Okay, they're also flexible, which allow them to be compressed by muscle action. And just an interesting fact, varicose veins are caused by weakening of the valves in veins. All right, now the heart is what pumps blood, but the blood that's going through the inside of the heart isn't dropping off any oxygen. Instead, the oxygen that supplies the heart cells as muscle cells comes from a branch off the aorta called the coronary arteries. Now, these are going to be arteries that provide oxygenated blood to the heart itself. Now, a note, epidemiology is the study of the nature and spread of diseases in the human population. We'll circle back to that in just a second. Now, these coronary arteries can become narrowed or blocked by fatty deposits containing a variety of lipids, including cholesterol. They restrict blood flow to this downstream end. Okay, often they cause chest pain, shortness of breath, especially during exercise. Okay, unfortunately, these fatty deposits, called plaques, can harden. They can make the inner surface rough, and that can trigger the formation of a blood clot, which is called thrombosis. Having high blood pressure increases the risk of that blood clot forming. What happens then if that blood clot forms, it completely cuts off the ability of oxygenated blood to travel past that point, and all of these cells that were served by that artery no longer receive oxygen and they die, and that is what we call a heart attack. The medical term is a myocardial infarction, often just shortened to an MI. Okay, so here we have done epidemiological studies, to try to figure out what are the risk factors for the common condition called coronary heart disease, which is when an individual has narrowed or blocked coronary arteries. And through those studies, multiple risk factors have been identified. Hypertension, smoking, eating too much saturated fat and cholesterol, obesity, high salt intake, drinking excessive amounts of alcohol, having a sedentary lifestyle, and then some things you can't control, like genetic predisposition and old age. Okay, note that you may be given some epidemiological data that you might be asked to identify trends on a DBQ, for example. Another note that correlation does not equal causation. This is just an important nature of science principle. This is why we never say, I've proved something, because we might always have new technologies that develop, new data that's collected, that's going to cause us to need to go back and assess what we know. And so we always say that we can identify a positive correlation, a negative correlation, or no correlation, but we never say with 100% certainty that something caused something else. All right, over to plants. Xylem is the tissue in plants used to transport water from roots to leaves. You can remember this because water starts with W and WXY. Those letters all go in a row. So xylem is a tissue that transports water. Now, this is driven by transpirational pull, where transpiration from the stomata up in the leaves pulls up all of these water molecules behind it against the gravity, without needing any ATP or energy input. just because of the cohesion and adhesion properties of water. Cohesion is when water molecules are attracted to each other because they're both polar. Adhesion is when those water molecules are attracted to other polar molecules like cellulose. And so those water molecules are going to be pulled up the xylem against gravity, okay, from root and then out through the stomata. And that evaporation, called transpiration, is what is just pulling all of that water up behind it. Let's take a closer look at the xylem vessels. Note they are wide, they have thick-walled, and they have these rings. Okay, xylem is non-living. There's no cell contents. Okay, these have incomplete or absent end walls. So you can see water is going to be able to freely flow up that xylem. Okay, but this is a popular question. Xylem has lignin. It's lignified, and that is an extra strengthening so that it maintains its shape whether there's high water pressure or low water pressure. It's extremely rigid. And we'll see in a bit when we look at a micrograph how thick those walls are. Okay, but they also have little holes called pits, which allow for entrance and exit of water. But this is a non-living xylem. Okay, we have here another drawing skill. You should be able to draw a cross-section of the stem. You should be able to identify each of these regions as well as what they're for. And then you should be able to describe within these vascular bundles. Okay, phloem is going to be towards the outside. That's where sugar is transported in the form of sucrose. And it's easy to remember that it's towards the outside of the vascular bundle because there are insects who will drink that phloem by inserting little proboscis into the phloem, okay, and be able to drink up all that sugar as their food. And so the phloem is going to need to be towards the outside so those sugars are available for those little insects. So that's just one way to remember that phloem is towards the outside. Okay, xylem, transport of water, is towards the inside. And then this cambium layer is just where more xylem and phloem are produced, and together we call this a vascular bundle. Okay, so here's that micrograph I promised you. Note that in each of these vascular bundles, it's very clear where the xylem is with those thick lignified walls. So all of these white ovals with the thick walls are your xylem tissue, again, towards the inside, towards the pith. Okay, this is another drawing skill. You should be able to draw a transverse section of a root. Here you can see that we similarly have the epidermis, and we now have an endodermis, the cortex separating them. Okay, the xylem is going to be clustered. You can draw this in like a 5-star shape like this or a 4-sided shape like a T. Then we're going to have clusters of phloem tissues. Okay, again, this is a plan diagram, so it's just not going to show individual cells, but rather regions of tissue. Okay, so let's compare that to an actual micrograph. Once again, we can see that xylem tissue with that lignified wall, those dead cells that are transporting water through, in this case, the root. Okay, this is the end of SL content for the whole video. Okay, so SL students, you guys can go ahead and turn this off. HL students, we still have the rest of this slideshow as well as the B3.3. So we still got quite a bit to go. Hang tight with me. All right, HL students, in addition to knowing about arteries, capillaries, and veins, you need to know about the connections, okay, between arteries and capillaries. And arteries, you need to know, branch into arterioles, just like we talked about bronchi branching into bronchioles. Okay, after the capillaries then, we have an intermediate as well on the other end, where the capillaries gather into venules, which then drain into veins and get returned to the heart. Now, the liquid part of the blood is called plasma, and that blood plasma consists of a lot of great things. Lots of dissolved substances, glucose, amino acids, mineral ions such as chloride and sodium, vitamins, hormones, plasma proteins. Lipids are carried in lipoprotein droplets. So we know that at the capillaries, we're going to need to do exchange of materials. And so with all of the neighboring cells, that blood plasma is going to get filtered out due to the high pressure on the arterial end. When it's filtered out, everything goes with it except for the proteins. because that's just too big to fit out as it gets filtered. Once it comes out, it's called tissue fluid. So tissue fluid is just blood plasma, the liquid part of the blood, minus the proteins that are too big. Okay, then we're going to see that tissue fluid circulates around all of those nearby cells that are near that capillary, dropping off oxygen, dropping off glucose, amino acids, and picking up CO2, urea, waste products. Now, most of that tissue fluid re-enters the capillary, but not all of it. Some of it is now called lymph and drains into these lymph capillaries, okay, lymphatic vessels, okay, and those are then going to join together, and they're eventually going to merge with veins and return that liquid to the blood system. All right, mammals have a double circulation system. Let's trace the flow of blood. So first up, we'll start here. You can start anywhere. It's a cycle. Okay, we're going to start here with deoxygenated blood. It's going to enter the right side of the heart through a vein. Veins go in. Then it's going to get pumped to an artery. Arteries go away. Okay, and it's going to then go to the lungs. At the lungs, it can get oxygenated. Following the lungs, it's going to now return to the heart through a vein. It's going to go in through a vein. The left side of the heart then pumps away from an artery. Now note this is now oxygenated. It's showing up as red here in our picture. Okay, note that one time the vein was blue, one time it was red, and one time the artery was blue, one time it was red. Whether it's a vein or artery has nothing to do with oxygenation. It just has to do with whether it's going in or away. Okay, that oxygenated blood then travels throughout the entire body, drops off that oxygen, and then returns on its path to the right side of the heart to start all over again. Okay, now we're going to see that the left side of the heart is actually significantly larger than the right side. The right side of the heart is pumping blood just a very short distance to the lungs at relatively low pressure because we don't want those alveoli surrounding the lungs to burst. On the other hand, the left side of the heart is much larger because it has to pump blood at high pressure throughout the entire body. This blood has to go up to your head against gravity. It has to go down to your toes and then back up against gravity. So this is going to come out at significantly higher pressure. Now, fish have a single circulation system. How do they get away with that? Well, fish live underwater, and so they can just pump the blood once at high pressure. They don't have to worry about us pumping at low pressure to the lungs because we want to prevent capillaries bursting in our 1 atmospheric pressure that we live in here on land. Fish live underwater. They're living under all that water pressure. That pressure is pushing in on them, which prevents the gill capillaries from bursting. And so they can pump at high pressure to their gills, and it's still at high enough pressure that it can then circulate through the rest of their body. And so therefore, they just have a single circulation system. Okay, let's talk about all of the parts of the heart. Now, we'll begin in the same place as when we talked about double circulation. First up, blood is going to enter as deoxygenated blood through the vena cava, singular vena cava. There's 2 of them. They're going to drain that deoxygenated blood into the right atrium. The blood is then going to move into the right ventricle through the atrioventricular valves. Okay, this is the general term for this valve that connects the atrium, there's the atrio part, with the ventricle. Okay, you can remember this one's the ventricle because it's shaped like a V. Okay, atrioventricular valve. Okay, the atria are going to contract together, followed by the ventricles. So when the ventricles turn to contract, okay, it's going to push that blood back upwards. That's going to cause the AV valves to close, and that blood's going to travel through the semilunar valves, okay, into the pulmonary artery. This is an artery, it's going away, and pulmonary refers to the lungs. So you can see it's actually going to branch immediately because it's going to go 1 to each lung. Okay, this semilunar valve is going to prevent backflow of blood flowing back into the ventricle from the artery. Okay, so now we've headed over to the lungs, we've gotten oxygenated, and it's time for that blood to come back to the heart, but now on the left side, which is completely separated by the septum. blood from the right and the left side of the heart do not mix. Okay, so here we have now oxygenated blood entering through the pulmonary vein, the vein from the lungs. It moves into the left atrium, through the AV valves, into the left ventricle. Okay, when the ventricles contract, it goes up here, semilunar valve, and then the aorta. The aorta is the super important artery that's carrying oxygenated blood away from the heart and towards all the tissues in the body. So you're going to see that the aorta branches almost immediately because that oxygenated blood has to go up to your head, down to your toes, out to the tips of your fingers, etc. So, a few adaptations of the mammalian heart for delivering pressurized blood to the arteries. Okay, so note the ventricles have a strong muscular wall. This dark red part here, that's all cardiac muscle. And so that contraction is going to be really strong and able to push that blood throughout the whole body. Atria, on the other hand, are going to be much thinner. They're just collecting blood from the veins and just have to contract it just right there down to the ventricle. Okay, so these AV valves, atrioventricular valves, prevent backflow of blood from the ventricle up to the atria, and the semilunar valves prevent backflow from the arteries, either one, back to the ventricles. Okay, now we're going to see that muscle cells in the heart called cardiac muscle are branched cells because that's going to more easily allow for electrical signals to be passed along those cells. And we're going to see that the heart is controlled by electrical signals controlled by the sinoatrial node, often called the pacemaker, which is located in the right atrium. It's going to send out a signal that causes the right and left atrium to contract together, followed then by the right and left ventricles to contract together. All right, it's that time of year. If you're about to do a promposal, here's a little note you could send to your desired date. Are you my sinoatrial node? Because you make my heart pump. Okay, the sinoatrial node is sending that electric signal. It will tell the heart when to speed up, when to slow down. Okay, right before you ask this other person, it might speed up a little bit, but who could say no to that one? All right, so again, the septum is this division between the two sides. It prevents those deoxygenated blood from mixing with our oxygenated blood. Okay, and then here's our coronary arteries and veins that are supplying the blood to the heart itself and then removing those waste products, the CO2 and urea from those cells. Okay, lastly, stages in the cardiac cycle. So the cardiac cycle is going to include the contraction of the atria followed by the contraction of the ventricles. Okay, and that's one cycle. It's going to be stimulated by the sinoatrial node, sending out that electrical signal. Okay, there's going to be a time delay of about 0.1 seconds between the atria contracting and the ventricles contracting. Okay, and we have two terms you're going to want to remember. Diastole is filling of the heart chambers with blood, and systole is contraction. So let's take a look at the cardiac cycle. Okay, as I outline these, I'm going to have you follow along, look at what's happening here with the pressure. Okay, remember that as something contracts, that increases the pressure. Okay, and to follow along with when our AV or SL valves are opening and closing. Okay, so that very first 0.1 seconds, okay, is atrial systole. The atrium contracts, causes a rapid but relatively small pressure increase that's going to pump blood from the atrium to the ventricle through the open AV valve. When we do this, the semilunar valve is closed, right? We don't want blood to be pumping up to the artery yet. We're just filling up the ventricle. Okay, so the blood pressure in the artery is going to gradually drop to its minimum as blood flow continues along it and no blood is being pumped in from the artery. Then from 0.1 to 0.15 seconds, this little tiny bit here, we're going to have ventricular systole. The ventricle contracts, a rapid pressure builds up, causes the AV valve to close to prevent that backflow to the atria. The semilunar valve remains closed until that 0.15 seconds to 4 seconds. That ventricular systole, the pressure in the ventricle rises above the pressure in the artery. That's going to open the semilunar valves. Blood is going to be pumped from the ventricle into the artery. That's going to maximize the arterial blood pressure at that point, okay, which you can see in this red dashed line, Pressure slowly rises in the atrium as blood drains in from the vein and the atrium fills. Okay, then from 0.4 to 0.45 seconds, we have ventricular diastole. The ventricle stops contracting. Pressure inside it rapidly drops below the pressure in the artery. So the semilunar valves now close. AV valves remain closed for that 0.05 of a second. And then we have from 0.45 to 8 seconds our ventricular diastole. Pressure in the ventricle drops below pressure in the atrium. So now our AV valve is going to open. Blood from the vein drains into the atrium and from there into the ventricle, causing a slow increase in pressure, and the cycle begins all over again. Okay, now HL back to plants. So we learned with the SL students that transpirational pull is what brings water up the xylem against gravity, no energy input needed, just due to cohesion and adhesion and transpiration, that evaporation of water from leaves. But there are certain circumstances that transpiration may not be occurring. Perhaps there's very high atmospheric humidity. If there's so much water vapor in the air, okay, you may not actually have any concentration gradient, and so then transpiration doesn't happen. Okay, we might have nighttime closure of stomata because we're not doing gas exchange, but those leaves still need water. Okay, or in the early spring, we might have like buds or small leaves on the deciduous plants that haven't opened yet. So they're not able to do transpiration and pull water up, but they're growing buds, growing leaves, and so they need water. So in all of these circumstances, we still need to bring water to the leaves, but it's not being pulled up because we don't have transpiration occurring. So what's going to happen instead of the free process, so to speak, of transpiration, we're going to need to input some energy, and we're going to use what we call root pressure. This is a positive pressure potential. It's going to cost ATP. Okay, and here's how it works. The root cells adjacent to xylem vessels are going to use ATP energy to pump mineral ions into the vessels by active transport. So we're going to increase the concentration of solutes using ATP. Now we know that if we increase the concentration of solutes in the xylem, that water will then follow by osmosis passively. Water always moves from low to high solute concentration. Now we discussed how the xylem have those lignified, rigid walls, and so we're going to see that water moves in, okay, and has nowhere to go except up the xylem. And so that root pressure is going to push water up the xylem, okay, and do what transpiration would have done, but just requiring ATP energy. Now, when we talk about the movement of sugars, instead of saying from leaves to roots, we have to say instead from source to sink. Oftentimes, the source is the leaf and the sink is the roots. The source is where the sugars are made, and the sinks are tissues that need to be supplied with those sugars. But you should know, the reason we have to say from source to sink is because sometimes the sink is above the source. For example, here we have some developing buds. Those are going to need sugars because they're too small and they're just growing and not able to produce enough sugars yet. So in this case, we might have sugars move through phloem tissue up the plant, okay, or down the plant. You'll never have both directions happening at once, but either one way or the other. So source to sink. Okay, so let's contrast what we learned about xylem earlier with phloem. Phloem is the tissue that transports sugars, and instead of being non-living, it's living. Okay, it has cytoplasm, it has membrane, but it doesn't have a nucleus or any other types of organelles. How does it stay alive? It has these little companion cells that are like life support systems with lots of mitochondria. And they are going to provide through these plasmodesmata all of the ATP and other things that this phloem cell needs to stay alive. Okay, now this is an adaptation then that allows for easy movement of sugars up and down these phloem sieve tubes. Now, these sieve tube elements are separated by sieve plates. These sieve plates have large gaps, you can see here in this micrograph, allowing for easier movement of those sugars. Okay, but Okay, they don't have lignified cell walls. They're just ordinary cellulose cell walls. And so the sieve plates also have the role of providing a little bit more structural support in the phloem because they are still connected. So they allow for easy transport while also providing structural support. Okay, here's an actual micrograph of, here's a couple of these sieve tube elements separated by a sieve plate. And then each of these black arrows is pointing to a helper companion cell, okay, that is helping those sieve tube elements to stay alive. Okay, so again, we are going from source to sink, but only one direction at a time. But note when we get to the sink cell, so say this is like moving from a leaf to like a root where starch is being stored. Say it's a potato. Okay, we're going to need to use active transport, okay, to pump those sugars into that potato where there's already lots of sugars and stored starches. Okay, but in doing that, then osmosis is going to move water into adjacent xylem vessels, which then can continue transpiration. Okay, and this is just a quick summary of that. All right, we're not quite done yet though. We have one more fairly slightly faster slideshow. But as a reminder, this is an all HL slideshow. So hopefully SL students have all started doing more practice problems and are not still watching, unless they just really want to know more science, which, hey, that's cool. All right, one of the things that all living things do is movement. Now you might be thinking to yourself, hang on a second. Things like trees or barnacles don't move, but movement could be locomotion where you move from one place to another, or movement could be within the body of an organism, just like we saw with all of that phloem moving sugars throughout the plant. Now, within locomotion, okay, we have those two categories of either modal, where the organism moves from place to place, okay, or sessile, where the organism remains in a fixed position, like most plants, but also some animals such as corals and barnacles are sessile. Now, for those who are using muscles to do locomotion, let's look at the anatomy of a muscle fiber. And there, look, you can already see that they are those multinucleated cells, which we've talked about before. So, muscle fibers are made up of many parallel myofibrils. Those myofibrils, if we zoom in, are made up of a series of sarcomeres. The sarcomere goes from Z disc to Z disc, and you're going to see they're named that because of this zigzagging shape. And on each point of the zig or the zag, we're going to see that there's an actin filament, a thin actin filament that is stretched out from the Z disc, okay, but it's fixed to it. And then we're going to see these thicker filaments in purple called myosin. Okay, and we're going to see that this is a contractile unit. That myosin is fixed in position, and these Z discs can move closer together as the actin slides along the myosin like intertwining fingers, okay, towards the center of the sarcomere. Now, if we looked at that under a micrograph, it would look like this. Here's our Z discs. Here's our thin actin filaments that go all the way into intertwining with the myosin, okay, part way. But when the muscle contracts, those Z lines are going to move closer towards each other. This large white gap is going to get smaller because more of that actin is now in between all of those thick myosin filaments. And so you should be able to identify this as the contracted muscle. Okay, so here's just sort of an image of what's happening when we see that contraction. Those myosin filaments have little myosin heads, and they're going to kind of walk their way up and pull those actin in towards the center of the sarcomere and therefore contract that sarcomere. Okay, and they do that, they form a cross bridge with the actin. Okay, and let's look at the actual mechanism for how that little sort of sliding, that walking occurs. Okay, it's often kind of compared to like a bunch of rowers kind of rowing their boat and pulling, in this case, sort of the water in towards the center of this sarcomere. All right, so let's look at the actual mechanism by which those actin filaments slide along those thick myosin filaments. Okay, so this represents our actin filament, this thin red line here. Okay, this is our myosin head in yellow. Now, this is our cross bridge that has formed. ATP can bind to the myosin head, which causes that cross bridge to break and the myosin head to detach from the binding site. ATP gets hydrolyzed to ADP and phosphate. This causes the myosin heads to change their angle. They get cocked up into their new position, okay, as they are now storing potential energy from that ATP, okay, but they're moving towards the Z line, okay, because now when they reattach, they're moving further out. They've basically pulled back and towards the Z line so that when they now pull forward, they can contract the actin towards the center of the sarcomere. Okay, so here those heads attach to the binding sites that are further from the center of the sarcomere. The ADP and phosphate are then released, okay, which allows for the power stroke to happen, where that myosin head now pulls the actin filament towards the inside of the sarcomere. So this is the actual mechanism, and this can happen repeatedly, okay, depending on what signals are coming from the nerve cells telling those myosin heads to keep pulling those actins in as the muscle contracts. Now, here we have the largest polypeptide discovered so far, called titin, indicated by this orange little spiral. Its job is to hold the myosin filaments in the correct position. It also prevents overstretching of the sarcomere, okay, but it acts also like a spring and adds to the force of contraction by releasing energy as it recoils. Okay, so many of the muscles in the body are antagonistic, like we talked about earlier with the lungs. A classic example of this is the bicep and tricep. The bicep muscle, when the arm is flexing, okay, it contracts while the tricep relaxes, and when the arm extends, the tricep contracts and the biceps relaxes. They're antagonistic. All right, skeletal muscles are composed of those striated muscle fibers, and they're stimulated to contract by a motor neuron. So this motor neuron is often to have many neuromuscular junctions with many different muscle fibers. That way, a single action potential that's being sent can stimulate many muscle fibers at once to contract, all in sync, so they contract at the same time, and then they can actually have enough contractile force to move. All right, so in terms of skeletons, there are both exoskeletons on the outside and then muscles on the inside, and endoskeletons like we have, where our skeletons are on the inside surrounded by muscle on the outside. Okay, let's talk about joints. Fixed joints are, for example, in your skull. When babies are born, their skull hasn't completely fused together. Okay, this helps with the birth process because it can kind of mold and move the baby through the birth canal more easily. Okay, and then within a short period of time after birth, the fusing of those parts of the skull occurs, but it's why you have to be so careful with a newborn, and especially with that soft spot right up at the top until the bones of the skull do fuse together. Okay, but most of our joints are joints that allow for bones to move, called articulation. Okay, and most articulated joints are called synovial joints because they have some synovial fluid, which acts as a lubricant, which allows for those joints to move with low friction. And yes, so you're not required to name muscles and ligaments, but you should be able to name the femur, okay, which is the bone that goes in your upper leg, that one large bone there. Okay, as well as the pelvis, which is where your hip bone right here is going to, your hip joint, I should say, is where your pelvis meets your femur. All right, so a few vocab words. Bones provide an anchorage for muscles and ligaments. They guide the types of movement that can occur at a joint. Cartilage is tough tissue that covers the bone at the joint. It also helps to prevent friction by preventing contact between regions of bone that might otherwise rub together. Okay, and it also can absorb shocks that might cause bones to fracture. Okay, so remember that synovial fluid I mentioned is a lubricating fluid. Okay, it fills a cavity in the joint between those cartilages on the ends of bones. Okay, it helps prevent friction. Ligaments are tough cords of tissue containing large quantities of the protein collagen. They prevent movements that would dislocate or damage the joint. Okay, so the joint capsule is that tough ligamentous covering to the joint, okay, that seals the joint and holds in that synovial fluid and helps prevent dislocation. Muscles provide the forces that cause movement at the joint. And lastly, tendons attach muscle to bone. Okay, like ligaments, they're composed of living tissue, with large quantities of extracellular collagen, which has high tensile strength. Okay, two types of joints. Hinge joints allow movement in one plane. For example, your elbow just goes in one direction versus, say, your shoulder is a ball and socket joint, which allows a wider range of movement. Okay, your hip or your shoulder. Okay, we've already discussed internal and external intercostal muscles, so we're going to go past that. Okay, and we're going to talk about some reasons that locomotion exists. It does require expenditure of energy for all that muscle contraction, and so we don't want to just spend ATP without any benefit. So what is the benefit to being able to move? Well, you can forage for food, escape from danger, search for a mate, and migration can occur. All right, now, while we know life began in the oceans, eventually some life evolved to live on land, and that's where we, for example, stayed. However, some living things then returned back to the ocean, and those are going to be our marine mammals like whales and dolphins. And they have some specific adaptations for swimming, okay, that because they're air breathing, because they eventually had ancestors that lived on land, we're going to see that they have many adaptations to allow them to be swimming marine mammals. So first up, they're streamlined. They're widest near the front and taper toward the rear, which causes less drag. Okay, they have that elongated teardrop profile, which reduces drag. Their body surface is smooth. There's no hind limbs or ear flaps, and they don't have any hair on their skin, which reduces friction. Okay, adaptations for locomotion. They have flippers used for steering in place of front legs. Flukes on the tail. Okay, those can go up and down and increase thrust. The dorsal fin to provide stability by preventing rolling. They have blubber, which provides buoyancy. Dolphins cease moving and float just below the water surface when they're sleeping. Okay, but they also have changes to the airways to allow periodic breathing between dives. Okay, they have a blowhole on the top of their head that leads from the larynx to the upper surface of the head. There's actually no connection between their mouth and lungs to avoid water entering their lungs. All right, that brings us to the end of B3. Hopefully that was helpful. Thanks for sticking with me.